Welcome, everyone. I'm Dr. John Burke from Rocky Mountain Cancer Centers, and I'm here with my partner, Dr. Chris Benton. We're going to have a discussion today about the latest and greatest updates from the annual meeting of the American Society of Hematology, or ASH, which occurred in early December 2022. Dr. Benton is going to tell us about some of the hottest issues in acute leukemias and myeloproliferative disorders. And so, Dr. Benton, let's go ahead and get started. Why don't you tell us what was new at ASH?
Thanks, John. I really appreciate it, and glad we're doing this again this year. ASH was particularly fun, I think, this year because a lot of us met there in person, and so it was better to be there at the meeting and actually get to see some of these things. And we have about 5 to 10 minutes today, and I'm going to run through three abstracts that really struck me as being forward leaps in some of the leukemias that we're trying to treat and develop better treatments for. The first one was abstract LBA1. LBA is a late-breaking abstract, and so this abstract, one of the late-breaking abstracts at ASH this year, was actually a long-term study that was initiated in 2013 for patients with acute lymphoblastic leukemia. These are adult patients with ALL, and the idea was in patients who did not have minimal residual disease, that were MRD negative after receiving chemotherapy, if the addition of blinatumomab was beneficial in these patients. And so the title of the abstract was Consolidation Therapy with Blinatumomab Improves Overall Survival in Newly Diagnosed Adult Patients with B Acute Lymphoblastic Leukemia in Measurable Residual Disease Negative Remission. Minimal or measurable residual disease is where we can't detect the disease after we give patients therapy. And so this was an ECOG Akron E1910 randomized phase 3 cooperative clinical trials network trial. This was presented by Mark Litzow, and senior author was Marty Tallman. So I'm going to just throw up the slide that they included in their abstract, which is available on the ASH website online. Let me share my screen here and see if I can pull this up. I don't know if you can see that, John, on the screen.
Yeah.
You're able to see that?
Yes.
So this was the way that the study was set up, where all patients received induction and intensification, and then they were tested for minimal or measurable residual disease. They had about 224 patients that were MRD negative out of some larger number of patients, 500 or so, that were treated initially. And so 112 patients were treated with blinatumomab, followed by consolidation, followed by a combination of blinatumomab and consolidation. The other 112 patients were randomized to receive consolidation only, and they all received maintenance. And they measured overall survival, and you can tell that these two lines are different. For those of you who haven't, are not familiar with Kaplan-Meier curves, this shows survival over a time frame, so months here on this axis. And what you can see is that there's a large separation between these lines, indicating that the group that received blinatumomab had significantly improved overall survival. And this really changes the way that we think about treating ALL. There have been a number of earlier studies that, in fact, one of the ones I presented last year at this ASH review that we did, John, from MD Anderson, was suggestive that the addition of blinatumomab was quite beneficial. And this is a randomized controlled clinical trial, which, really answers this question that blinatumomab with chemotherapy seems to be beneficial in patients with ALL. So that's the first abstract I wanted to show. I think it's a big step forward for adult patients with ALL.
So just to be clear, we've known that patients who are MRD positive should get blinatumomab, and now we know that we should also give it as part of their ALL therapy, even if they become MRD negative.
That is correct. So
Okay.
blinatumomab is approved for relapsed refractory ALL and is also approved for patients who have measurable or minimal residual disease after therapy. And now I think we can say that blinatumomab is probably beneficial in all ALL patients.
Got it. Thanks.
The second abstract I wanted to mention is abstract number 6, which was a plenary abstract in the scientific session. And this abstract was titled Discovery of INCA 033989, a monoclonal antibody that selectively antagonizes mutant calreticulin oncogenic function in myeloproliferative neoplasms. And, I mentioned this last year as well, but we're using more and more targeted therapies. And so this was the description of a monoclonal antibody that specifically targets CALR or calreticulin. Mutations in CALR are responsible for about a quarter of patients with myeloproliferative neoplasms like essential thrombocytosis and myelofibrosis. This particular mutant protein has a novel function that basically results in a constitutive activation, a constant activation of the JAK-STAT pathway. What they did was to develop an antibody that specifically targets the mutant calreticulin. And so these are a couple of the slides that, again, were presented and are available online. This particular antibody selectively inhibits cell proliferation and induces death of these leukemia cells, the cells that have this mutant CALR, which is shown here that at increasing doses in these preclinical studies, that this monoclonal antibody does quite a good job of inducing cell death. They even put this into a mouse model and showed that over time they were able to change the bone marrow and reduce the proliferation of megakaryocytes, which is characteristic of what we see in some myeloproliferative neoplasms. And so this is very promising. Monoclonal antibodies are quite tolerable, and these are already being used in phase 1 clinical trials in patients with mutant CALR myeloproliferative neoplasms. So I thought this was a noteworthy fact, and I wanted to spotlight it.
So these CALR mutated patients often have high platelet count and can get scar tissue in their bone marrow or fibrosis in their bone marrow. And this, in theory, may help, but it needs to be tested in human trials, right?
It's exactly right. Yeah,
Yeah.
but it's very promising from the standpoint of targeted therapy, where we're using a particular mutation that a patient might have and pairing up a therapy that specifically targets that mutation. I'll just mention the last abstract, which was abstract 63. It was an oral abstract on a menin inhibitor, also targeted therapy. And this is for patients with AML who have, KMT2R rearrangements or mutant NPM1. Again, specifically targeting AML patients that have these abnormalities. This is, again, a first in human phase 1 study, but it showed quite impressive results as a single agent in relapse refractory AML. And so I think I'll stop there and just, say again that these are targeted therapies that are promising for patients with these particular abnormalities. So I thought this was a noteworthy abstract as well.
Super. All right. Well, thanks so much for the update, and look forward to seeing these in the clinic.
Thank you.